हाँ प्लीज़।
आपने प्रेस किया है कुछ?
नहीं, मैंने कुछ नहीं किया। यहाँ से कुछ प्रेस हो रहा होगा तो बात अलग है, बट कुछ भी नहीं हो रहा। कहाँ चला गया सब कुछ? ओह शिट, कितनी सारी स्लाइड्स आ गई हैं। काम करिए, क्लोज कर दीजिए।
नहीं, ऑटो से है।
ओह शिट। अच्छा, लेक्चर के एंड पे वो कहता है एनोटेशंस रखना है या नहीं, तो वो कीप ही करना है ना? हाँ।
नहीं, डिस्कट कर तो भी चलेगा। कि
अच्छा,
आप जो लिखते हो ना
वो
वो
ऑटो
चले
सेव
जाता
पे।
है।
एक्सक्यूज मी,
कर दो चलो।
एक सेकेंड। कर सकते हैं।
टिश्यू है ना वहाँ पे?
सब है। As we have discussed about nucleotide chemistry, nucleic acid, that is DNA and RNA, today we'll be talking about purine metabolism. As nucleotide is made up of purine, that is nitrogenous base, pyrimidines, which is also a nitrogenous base, the pentose sugar, ribose and deoxyribose, and the phosphorus. So basically, nitrogenous base is synthesized in the body as de novo or via salvage pathway. The ribose sugar, which is present in the nucleotide, comes from the HMP shunt, and the phosphate group comes from the ATP, NADPH, and other sources of phosphorus. So, we'll be looking at the synthetic pathway of purines, that is adenine and guanine. As I said, the synthesis of purine synthesis happens via two processes. One is de novo, that is new synthesis, and the other way is salvage pathway. So in the salvage pathway, what happens that the protein which is sorry in the salvage pathway, the purines which are released during the degradation and digestion, or the purines which we consume in our diet, enters the salvage pathway for the synthesis of nucleotides. So basically, the purines are reutilized, and the purines which are present in diet is utilized via salvage pathway. So first we'll be discussing the de novo pathway for purine. Now let us look at the structure of the purine. As we know that purine is a bicyclical ring structure. So now we'll talk about the sources of various atoms which are present in the ring structure. So the first nitrogen atoms comes from aspartate. Second carbon atoms comes from N10 formyl tetrahydrofolate. Third nitrogen atom and ninth nitrogen atom comes from the amide group of glutamine. Fourth, fifth carbon atom and the seventh nitrogen atoms comes from glycine. Sixth carbon comes from the respiratory carbon dioxide, whereas eighth carbon atom comes from the N5N10 methynyl tetrahydrofolate. So to summarize this, the amino acid which are involved in purine molecule is aspartate, glycine, and glutamine. So sometimes you can be asked about the MCQ, that the amino acid which does not contribute to purine. Okay, so the one which will be other than aspartate, glycine and glutamine will be the answer. Tetrahydrofolate N5, N10 and N10 formyl tetrahydrofolate is involved in purine molecule, and other source is respiratory carbon dioxide. So purine molecule is made up of amino acid, tetrahydrofolate and carbon dioxide. Now, whenever we talk about any synthetic pathway, the first thing that we need to see is what is the substrate which is available for the synthetic pathway, the organ where it is taking place, and the cell organelle where the synthetic or the degradation pathway is taking place. So, for purine metabolism, the organ which is involved in the synthesis is liver. It happens in the cytosol and it does not occur in the RBC and brain. Remember that it does not take place in the brain. So, in the brain, the pathway which is responsible for purine synthesis is salvage pathway, both in RBC and brain. Now we'll look at the pathway first and then we'll discuss each steps in detail. So I would request you all to note it down so that you can add on to the further information of every step. Alpha D ribose 5 phosphate, which comes from the HMP shunt, gets converted to PRPP in the presence of PRPP synthetase enzyme. In the presence of ATP, utilizing two molecules of phosphorus in the presence of magnesium as a cofactor. PRPP gets further converted to beta 5 phosphoribosyl amine in the presence of enzyme PRPP glutamyl amido transferase. This step is a committed step and utilizes glutamine and converts it into glutamate, giving one molecule of amide. Beta 5 phosphoribosyl amine in the presence of synthetase enzyme forms glycinamide ribosyl 5 phosphate, adding glycine molecule to the substrate. Glycinamide ribosyl 5 phosphate in the presence of enzyme formyl transferase forms formyl glycinamide ribosyl 5 phosphate, converting N5N10 tetrahydrofolate to tetrahydrofolate. This FGR5 phosphate in the presence of synthetase enzyme utilizes one molecule of ATP to form 5 amino imidazole ribosyl 5 phosphate. 5 amino imidazole ribosyl 5 phosphate in the presence of carboxylase utilizing one molecule of carbon dioxide forms 5 amino imidazole carboxylate ribosyl 5 phosphate in the presence of synthetase enzyme utilizing aspartate molecule and one molecule of ATP forms 5 amino imidazole 4 succinyl carboxamide ribosyl 5 phosphate in the presence of enzyme adenosuccinate lyase, also called as adenosuccinase, releases one molecule of fumarate to form 5 amino imidazole 4 carboxamide ribosyl 5 phosphate in the presence of enzyme formyl transferase, releasing tetrahydrofolate to form imidazole 4 carboxamide ribosyl 5 phosphate, which further is acted upon by enzyme cyclohydrolase, releasing one molecule of water to form IMP, that is inosine monophosphate. So this is about the purine synthetic pathway. Now we'll discuss every step in detail. Now we'll look at the first step, that is alpha D ribose 5 phosphate gets converted to PRPP in the presence of enzyme PRPP synthetase. This enzyme is X-linked enzyme and it requires magnesium as a cofactor. It utilizes two molecules of ATP, that is two phosphorus molecule is added to ribose 5 phosphate, as you can see in the structure here, and this enzyme is activated in the presence of phosphorus and inhibited by nucleotide. The ribose 5 phosphate here comes from the HMP shunt, and this ribose 5 phosphate undergoes nucleoside phosphorylasis in the presence of PRPP synthetase. Now, before we get into the depth of the purine metabolic pathway, the first thing that we need to know in the purine synthesis is that ribose 5 phosphate is the primary compound to which the formation of purine takes place. Now, when we'll be discussing purine synthetic pathway, we'll realize that ribose 5 phosphate is added during the process, but it is not the primary component which is utilized. So this was about the first step. Now we'll go into the second step, that is PRPP. Now we'll go into the second step. Now we'll go into the second step in which this PRPP this PRPP leads to formation of 5-phospho beta D ribosylamine. So as you can see that this PRPP in the presence of glutamine and water releases two molecules of phosphorus which was added in the first step, leading to formation of 5-phospho beta D ribosylamine. So the amide group is added to the ribose 5 phosphate and the two molecule of phosphorus which were attached has been removed, leading to formation of 5-phospho beta D ribosylamine. Different books have different nomenclature in the purine synthesis, so I have ensured that the two names is mentioned in the slides so that whenever you read from different sources you should not be getting confused and realizing that both of them are correct. So 5-phospho beta D ribosylamine gets converted to glycinamide ribosyl 5 phosphate in the presence of synthetase enzyme, and as you can see into the amide group, the glycine is attached, one molecule of ATP is utilized and magnesium is used as a cofactor. Now this glycinamide ribosyl 5 phosphate in the presence of formyl transferase enzyme and utilizes N5N10 methynyl tetrahydrofolate, forming formyl glycinamide ribosyl 5 phosphate. In this step, the condensation takes place. Further, formyl glycinamide ribosyl 5 phosphate is converted to formyl glycinamide ribosyl 5 phosphate. To note that till the formation of formyl glycinamide ribosyl 5 phosphate, the ring closure has not occurred. In the presence of enzyme synthetase, utilizing glutamate molecule and amide group is transferred. Formyl glycinamide ribosyl 5 phosphate forms formyl glycinamide cut this one. So from glycinamide ribosyl 5-phosphate, formyl glycinamide ribosyl 5-phosphate is formed in two steps utilizing enzyme formyl transferase and synthetase. In the formyl transferase step, the N5-N10 methynyl tetrahydrofolate is utilized, whereas in the synthetase enzyme step, the ATP is utilized. This formyl glycinamide ribosyl 5-phosphate, in the presence of ATP and magnesium as a cofactor, releases one molecule of water, and finally the ring closure happens and the ring is formed in this step, that is in the conversion of formyl glycinamide ribosyl 5-phosphate to amino imidazole ribosyl 5-phosphate. This amino imidazole ribosyl 5-phosphate, in the presence of enzyme carboxylase, utilizes one molecule of carbon dioxide to form amino imidazole carboxylate ribosyl 5-phosphate. This, in the presence of synthetase, utilizes one molecule of aspartate, releasing one molecule of water to form amino imidazole succinyl carboxamide ribosyl 5-phosphate. This amino imidazole carboxamide ribosyl 5-phosphate, in the presence of enzyme adenyl succinase or adenosuccinate lyase, releases one molecule of fumarate to form amino imidazole carboxamide ribosyl 5-phosphate. Cut this one. So this amino imidazole succinyl carboxamide ribosyl 5-phosphate is acted upon this amino imidazole succinyl carboxamide ribosyl 5-phosphate is acted upon by enzyme adenyl succinase or adenosuccinate lyase, releasing one molecule of fumarate to form amino imidazole carboxamide ribosyl 5-phosphate. In the presence of enzyme formyl transferase, forms form amino imidazole carboxamide ribosyl 5-phosphate, and this component is acted by enzyme IMP cyclohydrolase, which leads to the closure of the second ring as well, releasing one molecule of phosphate forming IMP, that is inosine monophosphate. Okay, so now we look at the pathway in general. So as we saw that ribose 5-phosphate was the substrate around which the phosphate group were added using PRPP. Now that PRPP is added, there was addition of the amide group and release of the phosphorus group. So ultimately, what happens that ribose 5-phosphate was used as a base in purine metabolism, and further the ring closure takes place, and then further the closure of the second ring takes place in the last step, that is IMP cyclohydrolase step, leading to formation of inosine monophosphate. Okay, so this was about the formation of IMP. Now we're looking at the formation of other monophosphate. How IMP synthesizes AMP and GMP. Now look at the slide here. IMP, in the presence of enzyme adenyl succinate synthetase, from adenyl succinate. This adenyl succinate, in the presence of enzyme adenyl succinate lyase, forms adenosine monophosphate, releasing one molecule of fumarate. IMP, in the presence of enzyme IMP dehydrogenase, converts NAD plus to NADH plus, forms xanthosine monophosphate. In the presence of transamidinase, glutamate gets converted to glutamate, utilizes two phosphorus molecule to form GMP, that is guanosine monophosphate. So this is about the formation of AMP and GMP from IMP, which is the primary nucleotide formed from the purine synthesis. Now, how these monophosphate gets converted to triphosphate is via enzyme NMP kinase and NDP kinase. So, nucleoside monophosphate, that is AMP and GMP, in the presence of enzyme NMP kinase, that is nucleoside monophosphate kinase, utilizes one molecule of ATP to form nucleoside diphosphate, forming ADP and GDP respectively. Now, this nucleoside diphosphate is acted by enzyme nucleoside diphosphate kinase, utilizing one molecule of ATP to form nucleoside triphosphate. Now, to note here is that whenever there is utilization of, or whenever there is enzyme kinase, that means there is going to be addition of the phosphorus molecule. Okay? Now, how these ribonucleoside diphosphate gets converted to deoxyribonucleoside diphosphate. So this ribonucleoside diphosphate gets acted by enzyme ribonucleotide reductase, which in turn converts thioredoxin from reduced to oxidized form, and this oxidized form is further acted by enzyme thioredoxin reductase, which in turn converts NADPH to NADP plus, forming thioredoxin in reduced form, which gets further utilized in the ribonucleotide reductase activity, forming deoxyribonucleotide diphosphate. हनीफ, ये मक्खी निकालना पड़ेगा, कैमरे में आ रहा है.
ओ फैन भाई बैठे क्यों बैठे
देखो ना, पन्नी से ही निकल जाएगा, मुंह में ही आ रहा है. रिकॉर्डिंग में भी आ गया होगा शायद. निकल गया शायद. एक जो हटाया वो अंदर वाला ही था, आ गया फिर से अंदर. वापस इधर है, ये इधर 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 इधर
कुछ है देखो.
चला गया था यार वो बाहर.
चला गया.
नहीं नहीं, अंदर ही है, अभी अभी अंदर है, उस टाइम चला गया था.
क्या सुन है कुछ एक तो क्या
ये देखो, मेरे मुंह पे ही आ रहा है ये.
इधर इधर चलते बैठना इधर है, देख उधर देख, पीछे गया.
हाँ पीछे.
मैं तो
वो दो डोर पे है, डोर पे ये है रहा, ये रहा, ये रहा, ये रहा, ये रहा. इधर पे चला गया फिर लाइट के पास. वो फिर डोर पे एक है, देखो.
इसको दबाना. सर पे मत मार.
गया. प्लीज बंद कर दो जल्दी से. होपफुली गया. Now let us look at the regulation of purine metabolism. Ribose 5-phosphate is converted to PRPP in the presence of enzyme PRPP synthetase. This PRPP synthetase is inhibited by ADP and GDP. PRPP to ribosyl amine is Now, talking about the regulation of purine metabolism, ribose 5-phosphate gets converted to PRPP in the presence of enzyme glutamine PRPP amidotransferase, which gets inhibited by IMP, AMP, and GMP. And PRPP synthetase enzyme gets inhibited by ADP and GDP. Phosphoribosyl amine forms IMP as we have seen. This IMP forms AMP and GMP, and the enzyme which is involved is adenyl succinate synthetase, which gets inhibited by AMP, whereas this gets inhibited by GMP. So these are the inhibitors of or regulatory molecule of purine metabolism. And the enzyme involved is PRPP synthetase, glutamine PRPP amidotransferase, and adenyl succinate synthetase. Now we look at the salvage pathway. As I discussed earlier, that salvage pathway is basically a way of saving energy, whatever is utilized in the de novo pathway, as it is more energy consuming than salvage pathway. So the adenine which we get from the purine turnover or from the dietary sources gets converted to AMP in the presence of enzyme adenine phosphoribosyl transferase, utilizing molecule of PRPP, which acts as a donor of phosphorus group here in the salvage pathway. Guanine gets converted to guanine monophosphate in the presence of enzyme hypoxanthine guanine phosphoribosyl transferase, utilizing one molecule of PRPP, releasing two molecules of phosphorus. Similarly, hypoxanthine to IMP is via hypoxanthine guanine phosphoribosyl transferase. This also utilizes PRPP as phosphorus donor. Now, this enzyme, that is hypoxanthine guanine phosphoribosyl transferase, that is also called as HGPRT, the deficiency of which leads to Lesch-Nyhan syndrome, which we'll be discussing in subsequent slides. Lesch-Nyhan syndrome is because of the deficiency of HGPRT. So the conversion of Lesch-Nyhan syndrome is due to deficiency of HGPRT. लैपटॉप पे बैठा हुआ है मक्खी.
बुलाओ जो बेटा बुलाओ क्या वही
गया बाहर गया बाहर गया बाहर. Lesch-Nyhan syndrome that happens because of the deficiency of HGPRT enzyme, because of which the conversion of GMP does not take place and hence guanine is not converted to GMP. It is a sex-linked disorder. It occurs only in males. Since there is no conversion of GMP from guanine and there is no PRPP utilized, there is increased uric acid in patients of Lesch-Nyhan syndrome, which leads to gout. These patients also have mental retardation and learning disability, have nail biting tendency, which is called as self-mutilation. Lesch-Nyhan syndrome causes problems in the kidney. Due to increase in the uric acid, it leads to gout, and in the gout there is pain or deposition of urate crystal in the first great toe. It leads to CNS abnormality and also nail biting. Now look at this clinical case. A 5-year-old boy was brought to the hospital with chief complaint of aggressive behavioral developmental delay, learning disability, and irresistible urge to bite his own finger, that is self mutilation. Now, what is going to be the diagnosis? Now, remember that whenever there is enzyme deficiency or whenever there is any inborn error of metabolism, the first complaint the clinical case will have will be developmental delay, because these are present since birth, so child doesn't have normal growth. Second thing here, the characteristic feature of self mutilation. Whenever there is a word self mutilation, know that it could be Lesch-Nyhan syndrome, or it could be in porphyria. So the differentiating feature would be the CNS abnormality in case of Lesch-Nyhan syndrome, and in case of porphyria we'll get characteristic feature of photosensitivity on basis of which we'll be able to differentiate, and also in Lesch-Nyhan syndrome there will be increase in the uric acid. Okay, so remember that the diagnosis of clinical case is to be made on the basis of these characteristic features whenever asked. Now we'll talk about the degradation of purine. मक्खी है अभी भी, मेरे को तो तकलीफ नहीं है, कैमरे में आ रही है, मतलब मैं तो अभी ये देखो, ये देखो, मार दो यार, मेरे को मक्खी मारने की आदत नहीं है, मच्छर होता तो अभी तक मार देती मैं. ओह शिट, रिवेंज, रिवेंज फुलफिल्ड.
अब तू मक्खी रहेगी तो सब तेरा रहेगा.
तो अंदर उसको ऊपर ऊपर करो उसको, अंदर चले जाएंगे.
ऊपर ही थी, ये देखिए, इधर है, इधर है, इधर है, इधर है, इधर, ये देखिए इधर बैठी हुई.
क्या तुम मुझे खा दे रहे
यार वो एक मक्खी मूवी निकली थी तो उससे भी बुरा हो गया है अभी तो रिकॉर्ड करने के बाद.
क्या हुआ? मक्खी रहती नहीं, आज ये
आ जाएगी ना, मेरे को तकलीफ नहीं है, फिर आप लोग के कैमरे में आएगा बहुत क्लियरली, बस मुझे वही तकलीफ है और कुछ भी नहीं है मेरे को. जल्दी बंद करो, जल्दी बंद करो. मेरे को जल्दी रिकॉर्ड करना है, मैं लेट हो रही हूँ. Now we look at the degradation of purine. So purine, when consumed in diet, in vegetables or other dietary meat sources, the DNA and RNA which are present in mouth, because of the low pH, there is denaturation that takes place, and this denatured DNA and RNA, in the presence of this denatured DNA and RNA, in the presence of nucleases, which is released from pancreas, forms oligonucleotides. What it forms? It forms oligonucleotides. And this oligonucleotides is acted by enzyme phosphodiesterases, which is released from small intestine to form mononucleotides. These mononucleotides, by the release of phosphorus molecule, forms nucleosides by the enzymes nucleotidases. This nucleosides releases pyrimidines and purines, which enters back to circulation, enters salvage pathway, whereas few gets degraded to form uric acid or urea and gets excreted into the urine via kidney. Now look at the formation of uric acid. AMP is converted to IMP with the help of enzyme AMP deaminase. The deficiency of this enzyme leads to ADA deficiency disorder in children's. Okay. This AMP is converted to IMP. IMP is converted to inosine in the presence of nucleotidase. Similarly, nucleotidase converts AMP to adenosine, releasing phosphate molecule from AMP and IMP. This adenosine gets converted to inosine via again adenosine deaminase enzyme, releasing one molecule of ammonia. This inosine is further acted by purine nucleoside phosphorylase, that is PNP, leading to formation of hypoxanthine. This hypoxanthine is converted to enzyme xanthine. Xanthine gets converted to uric acid in the presence of xanthine oxidase. Now, xanthine monophosphate with the help of nucleotidase forms xanthosine, and this xanthosine gets converted to xanthine again via utilizing purine nucleoside phosphorylase. GMP forms guanosine via nucleotidase enzyme, which gets acted by purine nucleoside phosphorylase enzyme, leading to formation of guanine, and this guanine gets converted to xanthine via guanine deaminase enzyme, releasing one molecule of ammonia. So this is about the formation of uric acid from adenosine monophosphate, inosine monophosphate, and guanosine monophosphate. Now we look at the deficiency disorder in purine metabolism. First, as we have discussed, it is Leishman syndrome. Second, which is important, is gout. Third is ADA deficiency. Fourth is PNP deficiency. As we have seen that where these enzymes act and where these are utilized. So ADA and PNP and the enzyme because of which gout occurs is xanthine oxidase, is involved in the degradation pathway of purine. Okay. And Leishman syndrome is involved in the enzyme is involved in the salvage pathway. So, what happens in adenosine deaminase deficiency? This is an autosomal recessive disorder, and it is also a type of SCID, that is severe combined immunodeficiency disorder, which involves T cell, B cell, and natural killer cell depletion. Untreated ADA deficient children usually die within 2 years of age from overwhelming infection, and the treatment modalities which are present are bone marrow transplantation and gene therapy for ADA deficiency children's. Now we'll talk about PNP deficiency, which is also an autosomal recessive, and it is a very rare disorder which affects only T cell, and it is characterized by recurrent infection and neurodevelopmental delay. Gout occurs because of the xanthine oxidase cut. Now that we have seen formation of monophosphate to uric acid, now we'll look into the deficiency disorder involved in the purine metabolism. One is Leishman, which is from the salvage pathway, HGPRT deficiency. Second is gout, which is characterized by hyperuricemia. Third is ADA deficiency, which is because of the adenine deaminase deficiency. Third is PNP deficiency, which is because of purine nucleoside phosphorylase deficiency. These two disorder is a kind of autosomal recessive disorder. ADA deficiency causes SCID and children die within 2 years of age. In PNP deficiency, only T cell is affected, and it is also characterized by recurrent infection and neurodevelopmental delay. We have discussed Leishman, we have discussed ADA, and we have discussed PNP deficiency. Gout is something which is very important from exam point of view, so we'll be discussing in detail further on. Okay? Thank you. सो गाउट को एडिट कर दिया गया? हाँ, ये हो गया, दूसरा लेक्चर करना है अभी। हाँ हाँ, वो तो बंद हो गया, अभी दूसरा रिकॉर्ड कर रही हूँ। ये पहले वाले को ठीक किया क्या?
एक मिनट